Welcome back, everyone. So we have reached the end of our discussion of Chapter 7. We've been talking about the fluid mosaic model, and we're going to end by talking about the functions of those membrane proteins. Some really important membrane proteins are enzymes, and their presence in specific membranes is what accounts for specific functions being localized to particular areas of the cell or particular organelles. There are electron transport proteins, things like cytochromes and iron sulfur proteins involved in energy production. So you'll find things like electron transport proteins in mitochondrial membranes or in chloroplast membranes involved in those energy production steps. Membrane proteins are going to have important roles in uptake and secretion of substances, both endocytosis, bringing things into the cell by triggering a receptor on the cell, or by membrane-bound proteins creating that pseudopods that can create a vesicle to bring something in, or exocytosis by moving a vesicle towards the cell and fusing it with the cell membrane to allow things to be released from the cell. In the Golgi and the ER, where proteins are being targeted and sorted and modifying, there are proteins involved in doing that. There are proteins that detect light found in membranes. Some single-celled organisms that are able to detect light use those membrane proteins to detect light. And they also participate in autophagy. Autophagy is that process of a cell digesting its own self, its own broken down organelles or structures. So those are proteins, especially that what you would find in the lysosomal membranes, that are involved in digesting worn out or otherwise damaged structures or pieces of an organelle in that cell. So let's talk about a little bit more in depth about some of these functions of proteins. So transport proteins are proteins that move things in and out. A nutrient needs to come in, it's going to come in through a transport protein. There are channel proteins, something like this, that just provides a channel. It's a hydrophilic passageway. So hydrophilic things can't just go straight through the cell membrane. So by having this channel protein, there's a hydrophilic space where it's safe for charged particles or polar particles to come through. There are carrier proteins that are going to be more specific for a particular protein or a particular substrate to bind and be let through. It'll come, attach, and that triggers movement of the carrier protein and releases that substrate into the cell. And there are also transport ATPases. They are going to be transport molecules that require energy. So as they bring something in, it uses that energy from ATP to give it the energy to bring those ions across the membranes. And that's going to be more active transport, typically bringing things against the concentration gradient that the cell needs to bring into the cell. There are receptors. So here you have some sort of signaling molecule, a hormone or a growth factor or something that comes and binds to this receptor. This is a protein in the membrane. And when that receptor receives its signal, it triggers a change of other proteins. So here it triggers a change of an effector protein. Different types of receptors could bind to hormones, to neurotransmitters, or to growth-promoting substances, to growth factors. These are all different types of chemical signals that a cell could receive on its protein receptors. Cells also, as they are accumulating into multicellular organisms, are going to use membrane proteins to connect with each other. In animal cells, we have a protein called connexins that form gap junctions. So here's one cell, and here's the other cell. Cytoplasm of this cell, cytoplasm of this cell, and this is the extracellular space between the two animal cells. And these are the connexin proteins that are going to connect and form a gap junction so that these two cells can communicate with each other. And then here is plasmodesmata. In plant cells, we have plasmodesmata that connect the two plant cell walls and cell membranes to each other. This is a protein that allows the molecules within this cell's cytoplasm to be transported to this cell's cytoplasm. So those cells are able to communicate with each other. There are proteins that help give a lot of stability to the cell membrane and also shaping it. Things like actin. Here, actin is shown in red in this confocal micrograph. And you can see all of this around the cell membrane, around the edge of the cell. It's bright red because there are actin filaments right beneath that cell membrane, acting as part of that cell's cytoskeleton. There are also things like intermediate filaments that are helping stabilize the shape of that cell. So in all animal cells, we have this cytoskeletal meshwork of these different peripheral proteins. So this actin is not being incorporated into the membrane, but it is attached to the membrane just inside the cytoplasmic side. These peripheral membrane proteins that help underline that plasma membrane and stabilize it and shape it. The shape that actin takes, just like the skeleton of your body is going to give it the shape, this cytoskeleton is going to give it the shape of that cell. And these cytoskeleton proteins can actually rearrange themselves if the cell wants to push out a pseudopod. So here it's pushing out, or here it's pushing out. That's because we're extending those proteins to allow that cell to take a different shape. Many membrane proteins and lipids are glycosylated. Do you remember that word glycosylated? That means we're adding a sugar side chain onto a protein. So a lot of membranes contain some sugars on them. A glycoprotein, or a protein with a sugar, is going to be a membrane protein that has a sugar group covalently bonded onto that amino acid polymer. So when you add that carbohydrate side chain, that is what the process of glycosylation is. This usually happens to membrane proteins in the Golgi compartment. Sometimes it also occurs in the endoplasmic reticulum. Some of the common sugars we add onto a protein would be galactose, mannose, N-acetylglucosamine, and sialic acid. So you could have all of those sugars added to a protein, especially to the extracellular side of that protein, to act as a signal molecule to other cells. Glycosylation is going to involve adding a carbohydrate to either a nitrogen atom, such as here, a nitrogen atom of an amino group. That is going to be an N-linked glycosylation, N for nitrogen. That is going to happen if you have an asparagine as a side chain. If you have a serine, a threonine, or this hydroxylysine, which is a modified lysine, or hydroxyproline, which is a modified proline residue, these all have oxygens in them. So this is O-linked glycosylation. You're adding a sugar onto the oxygen, onto the O. So you can have N-linked glycosylation, or you can have O-linked glycosylation. But in the end, they're all adding different sugar molecules. Glycoproteins are prominent in plasma membranes, where they are important in cell-to-cell recognition. One you may have heard of is the ABO blood type. So these carbohydrate groups are on the outside of the cell membrane. So here you can see these sugars. These little circles are showing you the circles protruding on the outside of that cell membrane. And it's very easy to see how that could be a signal to another cell, that another cell could come and bind to that and recognize it and be able to create connections, or be able to verify that it is part of your body, or use that sugar as a way to say you don't really fit in and mount an immune response. Lectins are plant proteins that are very specific for the sugar groups they bind, and they bind those sugar groups very tightly and are used as a model to study glycoproteins. And the ABO blood group in people depends on differences in the different types of sugars. So here, the O-type blood doesn't have any extra sugars added to it. The A-type blood has this N-acetylgalactosamine added to it. That's what this little brown hexagon is. And B-type blood has galactose added to it. So if you have A-type blood, all of your blood cells have this brown marker. If you have B-type blood, all of your blood cells, or your red blood cells, have this blue marker. And if you're AB type, you have both. Some have you have both some molecules with the A and some molecules with the B. And if you're O type, you don't have either of these. So that becomes a way for your immune system to start recognizing your red blood cells as this is myself or this is not myself. And is also why when you get blood donations, the blood needs to be matched to your blood type, or it would activate your immune system to act as if there is some foreign body in you. A glycocalyx is something that we have in animal cells and sometimes even in bacterial cells. And the glycocalyx is the carbohydrate parts of the plasma membrane, glycoproteins, and the glycolipids forming this sugar layer. And that sugar layer is called the glycocalyx. So here, this is the glycocalyx of this cell. Here's the microvilli, and here's the cytoplasm underneath it. The glycocalyx is involved in cell-to-cell recognition, just like the other sugars we've looked at. It's also involved in adhesion. So if you think of sugar, it's kind of sticky. If you've ever spilled something like a sugary drink on your counter, sugar is sticky, so it's involved in adhesion. It's also involved in protecting the cell surface. So it gives it just kind of this little coat underneath it to protect the cell. It also helps create permeability barriers. Also, we saw earlier how lipids are very mobile. They're able to, we saw the experiment with photobleaching where lipids move really quickly and freely throughout the membrane. But a lot of proteins are more constrained. A lot of proteins are going to be kind of connected into larger protein complexes. They may be connected into complexes that go beneath the cell membrane into the cytoplasm that kind of constrain its movement, or connected into large islands within that membrane that is going to kind of constrain its movement. So while proteins are still able to move, there are some constraints and limits to that movement that we don't see as much with the lipids. We can do cell fusion experiments to be able to visualize how these membrane proteins are moving. So here's an example of what David Fry and Michael Edidin did. So they took a mouse cell and a human cell. This mouse cell is covered with mouse proteins, human cell covered with human proteins. And they treated it with the virus, which caused these two cells to fuse together. This is a property that some viruses have, is they cause cells to fuse together. So now they have this combination of two cells. The mouse proteins are labeled with a green fluorescent stain, and the human proteins are labeled with a red fluorescent antibody. So if the proteins aren't moving at all, you would expect the green to stay at one end and the red to stay at one end. But after allowing that cell to sit for about 40 minutes, you start to see there's red and green moving all along. Even within a few minutes, you can see the red starts to move up and the green starts to move down. So we do know that membrane proteins are able to move by seeing how the red and green labeled proteins are moving around on this cell. So to do this, Fry and Edidin used human and mouse cells that had different fluorescent antibodies. So the antibodies that fluoresced to the mouse, the antibodies that bound to the mouse proteins fluoresced green. The antibodies that bound to the human proteins fluoresced red. That stain that they used with the mouse antibodies was called fluorescein. That's the green stain. And rhodamine was the red stain that was used on the human antibodies, and gave this beautiful picture of fluorescing red and green where these proteins were moving. The diffusion of a lot of those proteins was restricted to a limited area of the membrane, and that is going to create a membrane domain. So a membrane domain is that specific part of the membrane that has a specific protein composition that's a little bit different from what you see in the rest of that cell membrane. And so that specific membrane domain is going to have a specific function. There are membrane domains that form structures that are barriers to diffusion. There are membrane domains where the membrane proteins will aggregate together in the membrane, and they'll form these large complexes that are kind of slow to move around. The most common restraint on the mobility of membrane proteins is anchoring them to structures on one side or the other. So it could be to they are being anchored to cytoskeletal proteins, so they're kind of stuck in their area. Or it could be that those membrane proteins are adhering or interacting with the extracellular matrix, which also is going to constrain its ability to move. Okay, I hope that chapter 7 was a little bit of a lighter chapter. We got through some kind of complicated signaling pathways in our last unit, and now a lot of this membrane should be more of a review of what you learned in your first biology classes, maybe a little bit more information, but nothing too big and mind-boggling to cover. We're going to talk about our membranes a little bit more in chapter 8, so we do have a little bit more to come visit, and I will see you then. Bye.